There you go. Right. So this is being, a session on development. I decided to push it a little close to very early, the earliest we could. And so one reason I love this as a first slide is because it shows beautifully that us as vertebrates have a very complex brain, but we're not quite as complex as the whale. So basically it also shows an important thing is that when we speak about size and complexity, it's size and complexity relative to the other part of our brain. So of course the whale has a much bigger brain than we have because it's a bigger animal, but what we're speaking about really is how complex and big is the forebrain here in green compared to the rest of the brain. And so when we speak about this, clearly the whale has a much more circumvoluted and folded forebrain that we have. And I attract the attention also to the fact that the two parts that are mostly variable across vertebrates are not only the telencephalon and mostly the cortex in the telencephalon, but also the cerebellum. Now, as I said, the big question is how early is our brain human? And it's almost a philosophical question, but I'm going to transform it for today in a much more down-to-earth question, which is when and how is telencephalic size controlled? We are used to think that the common views, at least, are that up to late somatogenesis, late somite stage, we vertebrates are all very similar. This is, of course, a famous drawing that's not the most accurate and rigorous drawing of that kind of stage across the vertebrates, but it does reflect this idea that we actually all pretty much look the same up to late somatogenesis, and after that, the forebrain is basically taking different type of decisions, and Arnold shows beautifully how neurogenesis and progenitor types are changing more or less from that time onwards. Now, of course, when we look at details and we look more carefully, we see differences as early as the neural plate. So, of course, our brain is first arising from a very simple sheet of cells that are closing up into our, the usual image that we have of our brain, and these are embryonic brains here, lateral view. But when you see these neural plates, you can see in these different shades of colors that the future telencephalon here as a horseshoe is having a different shape across from fish to mouse, with the fact that, again, relative to the other territories, the telencephalic field is taking more and more space into that neural plate. So the question is really, is this a coincidence, or is it that with evolution, the fact that we basically commit a bigger part of our neural plate to telencephalon is actually required for our telencephalon to become bigger later. So how much impact these variabilities have into the kind of forebrain we'll make later? So the questions are really, how is the relative size of the telencephalic field in these neural plate controlled? And then, is the size of the early territory influencing the size and complexity of the brain later? To answer this question, we actually, of course, needed to know more about the neural plate. And the next slide is a little bit of a nightmarish slide for you, which summarizes 10 to 15 years of work on the neural plate in the zebrafish, an exotic animal model that quite a few of us use to look at genetics of early brain development. And that basically organism allowed us to have a model of the different steps that leads to these allocation of territories inside the neural plate. Now, I don't need any of you to remember any of these details. I just put them all up here because some of the people in the room might be actually interested. But what I want you to remember are two really simple things. One is that the process of allocating this territory of telencephalic cells inside the neural plate is progressively happening in time. So there's a temporal clock that allows these to happen so that we have this horseshoe-shaped territory in the neural plate. And the other information that I want you to remember is that this allocation of telencephalic fate depends on two signaling centers. One located at the anterior neural border of the neural plate, that is called the anterior neural border, that is secreting Wnt antagonist, and another place in the neural plate that's more posterior, that's called the mid-hindbrain boundary, or MHB, secreting Wnt signal that basically is posteriorizing or imposing posterior identity to this plate. So Wnt activity would actually push towards diencephalic identity, and no Wnt identity will actually push into telencephalon or eye field. Eye field is because our retina is coming from the neural plate and budge out of the neural plate as the neural plate closes. So two signaling centers, anterior neural border and mid-hindbrain boundary. The anterior neural border might mature into what's known in the mouse as the anterior neural ridge, sitting at the late neural plate stage at the very edge and secreting FGF. Now, that's the case for fish. What about mammals? There are very few things known in mouse about the early stages, and these are summarized into this timeline. So this is now a timeline that is another kind of representation of the drawing you just saw. So mid-gastrulation arising of anterior neural border and mid-hindbrain boundary, setting up the identities in the neural plate, and then maturation into the anterior neural ridge, settling the cells into telencephalon fate. In the mouse, we know that there are a bunch of transcriptional factors expressed in the anterior neural plate that actually are found in the fish at equivalent stages. And we actually know that at four-somite stage, we begin to see in the mouse this horseshoe territory that is the future telencephalon, expressing FOXG1 and requiring the anterior neural ridge. Now, the first thing we did is to ask, is there an anterior neural border in the mouse? And we did that thanks to a collaboration with Anne Camus in Paris, who basically first fate mapped the anterior neural territory in the mouse at E7.5, E7, actually basically late bud stage, so it's more precise. Finding out that this territory here on at the very top here is purely ectodermal. There is an intermediate population that's a mix between neural and ectoderm, and then a proper neural territory here. And using this knowledge, she then did a series of ablation in the mouse embryos, and that's really tricky because you have to take the mouse embryos out of the mouse, do the ablation, and keep them in culture. You can, of course, not put them back in the mouse. The culture cannot be really good past 24 hours, so the stage at which you can look at is only around 8-somite stage, the latest. And she sees that when she ablates only this ectodermal territory here in the mouse embryo, she basically sees that she doesn't see anterior neural ridge, so she loses FGF, but she also loses any of the markers that are markers of anterior forebrain telencephalon in particular. So it seems that this population here is acting as an anterior neural border. Now, in the fish, we identified the anterior neural border because we knew that if we take it out of the fish, we don't make telencephalon. And if we actually put this cell population posteriorly in the CNS, in the posterior neural plate, future spinal cord or hindbrain or midbrain, you actually are able to induce ectopically telencephalic fate around that transplant. So we decided to do a crazy experiment, which is trying to find that whether we could take cells from the mouse embryo and basically replace the anterior neural border of the fish by the mouse. And ask two questions: can we replace it? And can we also see ectopic induction of fate if we use the mouse population in the fish? We had a proof of principle, which is actually transplantation of the node. I have no time to show you that, but basically it works. So we know that the mouse cells can survive in a fish and signal so that you can actually do a node induction, secondary axis. We decided to test a different population, the population that Anne Camus identified in the mouse, a population that's slightly posterior to that, much posterior to that, the lateral. And we found that these two populations can rescue the telencephalon. So these blue splotch here are telencephalic markers. And you see these two expressed in an embryo that is a zebrafish embryo without its zebrafish signaling population replaced by a mouse population. And you don't see this induction was the lateral population or the most posterior population. And this induction persists, so this is a confocal section of a brain, fish brain, where the red is showing you the telencephalic fate, it's FOXG1, and the cells that are actually mouse cells are spat out of the neural tube and actually create a little organizer on the side of the fish. Organoid, I mean. So it's a very small part of a telencephalon growing in the fish. The ectopic works as well. So when we transplant it ectopically, we have ectopic induction of telencephalic fate on the side here that you can see in this embryo. And the lateral or other cell population we tried don't do that. So it's specific to these two areas here. The most effective is the most anterior one, which actually persists into making this big extended telencephalon later on, whereas the one that actually act early, here, the green one, doesn't actually persist late in its fate transformation. So we have an anterior neural border in fish, in mouse, and we did a RNA-seq comparative that I don't have time to show you, but that shows actually 57% overlap in gene expression between the two populations. Now, what's the most important to show you is the following, which is that one of the striking obvious conclusion of these two timelines is that the anterior neural border and mid-hindbrain boundaries are actually appearing at the same time in fish, whereas they're temporarily separated in mouse. So using the zebrafish again, we were able to basically use transplantation to put an anterior neural border earlier in time in fish to have the equivalent of the mouse situation. And when you do that, you have a huge bulky telencephalon. So just temporarily changing the time at which you have the anterior neural border leads to a big brain. You don't have that if you increase the amount of neural border in a homochronic fashion. When you see what happens is that you have actually this transformation from neural plate onwards. So instead of what I was expecting, which is stretching anterior, including the eye field, you basically transform part of the eye field into telencephalic fate. That is followed by an increase of the ANR-FGF secretion, a huge population of secreting FGF, delayed neurogenesis 3 somite later, in a bigger number of neurons, and at the end, an amazingly pallial extension in the fully formed telencephalon, so more of the future cortical equivalent. So basically, when you do have an anterior neural signaling center arising early, you can actually move from a smaller telencephalic field to a bigger one, and that in turn snowball into a bigger telencephalon with a more pallial identity. This is shown also by mutation, and I don't have time to show that, but basically it's fairly obvious that that's what happens. Now, this is bigger, but is it different? Is it just a bigger version of a fish brain? And so we used our atlas, so we made a whole atlas of the zebrafish brain from 0 to 30 days because it didn't exist. So brain, no telencephalon in particular. And using our atlas, we actually looked at the brain at 30 days. And in these big zebrafish brains, what we see is that we see the arisal, appearance, the bone, the formation of a new set of neurons, because SADB2 is actually never expressed in the telencephalon in fish, and it arises in these big brains. So it's not only bigger, it makes different neurons. And so I think that this is telling us that the very early decision of making more or less cells in the neural plate has a huge impact of the kind of brain you're making, and therefore early really matters. We want to actually extend the study, not only comparing fish and mouse, but we want also to compare mouse and human. And of course, human embryos, we are privileged to be in England and having embryos from the clinic, but they are actually very rarely, if not never, early enough for us. And so what we do, and I don't have time to go on about this, but we can discuss it because I think it's important to discuss these things. We're actually embarking on making neural platoid by basically using bigger version of the macro pattern that was developed by Ali Brivanlou. And we actually induce breaking the symmetry of these discs by transplanting part of mouse nodes on these discs and look at what happens. And what we do is we basically obtain these cell population in which we have anterior part, which is 6-3 positive, and we have this weird heterochrony. So we have cardiac beating cells here. Of course, we don't have any blood, but these beating cells should actually not appear at these stages. This is 7 days after induction. So there are weird heterochrony in culture that is bound to be due to the cell culture, and we have to discuss this. Actually, it's part of the, organoids are strange. So we want to basically, this is starting from iPS cells, and that might be one of the issues. But we want to use this system to compare mouse and human, and I'm finishing there. I'm slightly late, so I'm going to avoid the conclusion, blah, blah, and do the really important thing, which is the work. Most of the work has been on Perry Müller, who did the work on the mouse versus fish A and B. Holger Billen has done the work on the heterochrony of the anterior neural border by transplant. And these 3 guys, fantastic guys as well, Florence, Isabelle, and Afnan, are continuing the work in my lab today. Thank you.
So I'm Rick Livesey. So Corinne, there's a slight contradiction on what you said. You said you basically take a neural field and you can eat into the retinal field to generate a larger encephalon, and then the retina does regulative development and looks normal at the end. So for retina, it's a different rule for pallium. Is that the kind of shorthand?
Yeah. Well, so we knew that. So we did ablation of retinal cells, and we don't find any difference in fish retinal size. But it's something that we expected, actually. So the expected thing would be that it wouldn't matter, because when you look at the retinal field in the neural plate across vertebrates, you don't see correlation between the retinal field and the final size of the eye. So the chip has a very small retinal field in the neural plate and a huge eye. So clearly the eye size, the final size of the eye, is purely controlled by progenitors later than neural plate stage. But the beauty of it is that the telencephalon doesn't seem to be the case, of course, because when we change it by mutation or by other means, we have a bigger telencephalon, we change the size of the territory, and it seems that it would actually be, it's a clever evolutionary mechanism if confirmed in other organisms, because it allows evolution to tweak the size of the telencephalic territory without affecting the visual system. So, you can think about it, and it's probably happening in other places of the neural plate. So, if you have territories that are regulated later and other territories earlier, you can tweak the fate between these 2 territories early and not affecting one of them. So the idea is that, it doesn't matter if you restrict the retinal field for the eye, but it matters for the brain.
Yeah, François.
So you are jumping from mouse to human to study the evolution of the neural plate and allocation of telencephalic territory. So is it because you think non-human primates are not appropriate, like marmoset, for example, where you would probably, it would be perhaps easier to get early stages? Are they good models? Arnold was arguing that maybe they are not ideal model to study outer radial glia. So how about these earlier stages?
Yeah, so we could do this. I was trying to, because the organoid field is booming and there are lots more tools, if we manage to have a good organoid model, we there is available early embryos for one cell that people would actually, not use, then we can look at it for sure. We also looked at the cichlid system as well amongst fish, and we see the same correlation of change in neural plate, translating to change in mature brain. But that's amongst fish.
Great. Oh, yeah, one last question, yeah.
Thanks. Kate Story. I was interested in the origin of the new cells that you see. They're definitely not from the graft. So the new neurons that are generated in your fish.
They're definitely not mice, yeah.
So they're definitely zebrafish. And
Yes.
the kinds of things that they express correlate to what kind of cell population in the mouse?
So the SAP-V2 telencephalic population would be upper layer cortical cells. The fish, the zebrafish, doesn't have a cortex and doesn't have layers. So it's, speaking of jumps, mouse to human, it's zebrafish to mouse is a big jump too. And so we cannot really make, daring claims about what these neurons are until we look at behavior. And so now we're building a genetic way of creating this big brain zebrafish so that we can actually look at how this correlates to behavior and where these neurons are connected to.
So I guess that was my next question, was about the behavior. So do these fish behave differently?
Yeah, so we didn't do that yet, mostly because it's all based on transplants and they are variable, and I don't like the variability that it generates, so the genetic way of doing it is the best route, and then we're going to go through behavior.
Thank you.
Great. Corinne, thank you very much indeed.